Please, sir.
Okay, let me share my screen. Yeah. Can you see me in full screen? Can you see me in full screen? I think there is some problem with sharing. Can you
Today,
see?
sir is going to describe a topic on understanding
Okay.
adaptive evolution of the
So
sensory
yeah,
microbes from healthy human
and
microbes.
thanks for this opportunity to talk about my research in front of the upcoming scientist or maybe uh, next generation of science. So I will be talking about all my work since my PhD in CSIR ISCB Kolkata, and then a postdoc in Germany. So I will be dividing my talk into maybe two portions. One is my PhD research, in which I developed three novel software for analysis of microbial genomes, and applied this software to understand the evolution of bacteria from human microbiome, which is the healthy human microbiome. In the third phase, I did antibiotic resistance profiling of this human microbiome and tried to find out body site specific gene functions in those microbiome strains. And in my postdoctoral research, I did same kind of evolutionary analysis with the symbiotic bacteria that are mostly found in environmental or lakes or groundwater, and then extended that study to study evolution, whether these bacteria are under reductive evolution or not. So to start with, first of all, what is human microbiome actually? So human microbiome is a set of all the microorganisms that are living inside or around our body, and these may include bacteria, archaea, viruses, fungi, and also eukaryotes. But mostly these are like 99% bacteria. So their complete genomic material functions and everything is collectively known as the human microbiome. And as we can see from the figure, that at every body site there are different set of bacteria that are present, and they differ in their functional properties. So what are the effects of human microbiome on our health? So it is known that they facilitate digestion for complex carbohydrates. Microbes inside our gut also synthesize some vitamins for us. They attenuate inflammation by releasing some like stimulating some anti-inflammatory factors, and they also modulate our drugs that we take as a medication. They also train our immune system to differentiate between good bacteria and bad bacteria, and they also protect us against pathogenic infections. So so all this is right, but do we have data for analyzing and studying these microbes or bacteria? So the human microbiome project was initiated by NIH USA, National Institute of Health. So they provide us data with different kinds of data, which is like 16S rRNA sequencing data, whole genome data from the metagenomes, and cultured bacterial strains and their genomes. So, to analyze this vast number of strains, there is a concept which simplifies the analysis, which is a pangenome concept. So, it is a simple concept. Assume that there are three bacteria or genomes, and you are studying them collectively. So, the pangenome will be the union of their gene set. So, genome A will contain, suppose, 500 genes, C B and C. So there will be some part which is like common in those bacteria, which is called core genome. There will be some parts where there is overlap. It is called accessory genome, and there will be definitely a strain-specific genome. So what we did was we did a statistical approach to model this into some kind of statistical model, which tells us that when we increase the number of genomes that we are studying, like 1, 2, 3, 4, the total number of gene families in the y-axis keep on increasing, because there will be obviously some overlap, but there will be strain-specific genes that will add to these numbers. And the core genome, which is common gene families, conserved gene families in the y-axis at the lower plot. As we add as many genomes, the number will keep on decreasing. So that's a norm. So these pangenomic approaches can be applied for various purposes, like designing vaccines. So it will provide a preliminary data for evolutionary analysis, and obviously habitat specific adaptive features of these bacteria. So we developed 3 novel software where we can analyze the multiple number of strains. It may go around thousands of strains from of bacteria using bacterial pangenome analysis pipeline, in which we use a complicated protocol to find out the conserved genome, the accessory genome, which is shared genome between 2 or more strains, and a strain specific genome. So, and we applied this software to a large number of like 1,293 strains coming from human microbiome project from different phyla of bacteria, and to create the resource, which is PanGFR-HM, which is pangenomic and functional resource. So it was an online tool. So right now it is being migrated to a private institute, because the PI, the scientist under whom this was like authorized, is moving to different institute. So it may not be available right now, but this is how it looked. So it analyzed more than 1,200 bacterial strains from human microbiome. And then a visualization software was developed to visualize the chromosome of some bacteria or some eukaryote as well. So it takes the genome file as an input, and the sequencing reads to align to that, to predict the various gene functions, and also align the genome synteny, like the position of each gene in all the studied strains. So these are all things that can be applied to a greater research. So first we focused on Priotella, which is a bacterial genus mostly present in a rural microbiome, and it is also highlighted in various studies indicating their role in the some of the diseases as well. So what we did was we used all the strains available from the Human Microbiome Project to model the same kind of thing that I discussed, the pan genome. So for this is practical data for 42 strains. So as we go from like the first strain to 42nd strain, the number of gene families, which is like overlapping genes, increases exponentially, or you can apply a power fit, so that you tell about the trajectory. So if the trajectory is too upwards, so we can call it as a open pan genome, and if it is saturating at the end, like this slope will be decreasing at towards zero, then that pan genome is called a closed pan genome. So what pangenome data can also be used to reconstruct the phylogenetic relationship between these Priotella strains from different body site. The color code tells you about their origin of body site, for example, airways, gut, oral cavity, and their interrelationships in the phylogenetic distance. So we also analyze the each and every strain where there are strain specific genes or not. So these 4 strains, 2 from gut and 2 from oral cavity of Prevotella, showed a high number of strain specific genes. So suggesting that it may not be so it may be a different taxonomical unit that is mislabeled as Prevotella genus. So, and next, what we did from the pangenome data, we tried to find out the body site specific genes. So, for example, all the green species of Priotella, they contain some genes which are not present in any other body site. So the vertical line means presence of a gene in a genome, and blank white space is just absence of that gene. So we found out the body site specific set of genes for each body site. And then there was some interesting observation where some genes were absent exclusively from that body site. So it might be a different angle of evolution where the bacteria lose genes to adapt well to the environment. So these genes were mostly carbohydrate metabolism genes, and many other categories. For example, defense mechanisms was very high in airways because we are continuously exposed to pathogens through air, and like so on. So there were also some genes or pathways that were absent. So most of the pathways that were absent from skin were broad metabolic pathways, but also biosynthesis of secondary metabolites. These pathways were not present in the skin Prevotella bacteria. So to conclude this, we say that Prevotella pangenome from human microbiome is open pangenome. So the genes that are conserved in this Prevotella genome are essential for their adaptation to the body sites. And along with the presence of genes, there are like some genes lost, or absence of genes is also important for adaptation to particular body sites. So, and from the phylogenetic tree, it was not clear, it was clear that there is no role of body site specific genes in the phylogenetic relationship, but they follow their traditional phylogeny, their species genus structure. So in the second step, we studied a second genus, which is a very commonly known genus, Lactobacillus. So many of them are also marketed as a probiotic preparations. So they are present in 3 body sites in human microbiome, and we tried to map this pangenome and to the Lactobacillus pangenome also. And as we can see that the trajectory is going upwards, so it is an open pangenome. So we also found out body site specific genes in the Prevotella. So as you can see, the blue ones, for example, this category N cell motility genes were very high in like vaginal lactobacillus. This is VL. So that might be important for their survival in a respective environment, because the pH, moisture, and other things also affect their stability. So there are some marketed preparations or like Lactobacillus rhamnosus GG strain. It is marketed as Enteroplus for probiotic effects, beneficial effects. So what we did was used all the lactobacilli genomes from the human microbiome to find out the features, like some gene features which are responsible for adaptation to human body, and also some of the probiotic factors by looking at the presence absence of the genes. So the simple clustering of these metrics of presence of genes responsible for different probiotic effects or health effects to human body, we highlighted that the marketed trend genome is clustered with 2 other genomes of Lactobacillus fermentum and reuteri, which have this high number of probiotic genes, and it can be a potential probiotic in future. So that's what I am summarizing here. Next, we looked at the antibiotic resistance profile of human microbiome bacteria. So what we did was we tried to find out the antibiotic resistant genes from all of these available genomes, to tell about presence of there are different categories of quinolones that you as a pharmacist might be knowing by heart. So sulfonamides, macrolides, quinolone derivatives, multi-drug resistance genes, and amino glycoside resistant genes. All these genes were mapped to each and every genome in the phylogenetic tree to show that the known pathogens show high number of this presence of antimicrobial resistant genes. The darker color of the square is indicating the high number of genes present. So all the Enterobacter, Klebsiella relatives of pathogens show antibiotic resistant genes. So what we did, expanded this to all the body sites, and we found out that the same pattern was present in Klebsiella from gut, airways, urogenital tracts, E. coli, and these are known pathogens, but they are also present in human microbiome. So these carry high number of antimicrobial resistant genes. And secondly, also the Enterococcus relatives, Enterococcus faecium and some other relatives also had this high number of genes. But the lactobacillus strains, as you can see here, did not have any of these strains from different body sites like urogenital tract and gut as well. So this shows that those strains are safe for providing as a probiotic or something. Then we analyze a large number of data from Human Microbiome Project, where we already actually have the whole samples of like DNA metagenome sequencing, and not just genomes, all the total metagenome. So so these 415 metagenomes are the environments or different body sites sampled and then sequenced for all the DNA present inside. So each dot here is one sample or from one person. It might be from oral cavity, gut, urogenital tract, or skin. But when we plotted it as a clustering algorithm, so every group is separated significantly different from each other. So each dot is indicating one person. So the group of oral cavity metagenomes were separated from all others, and likewise. So when we repeated this based on taxonomic abundance, based on presence absence of these enzymes in those particular samples, and presence also the relative abundance of those functional enzymes. Every time the groups were significantly differentiated from each other. We also found out the common core genome between all these samples. So we also plotted the relative abundance of these core genomes, means all the genes or enzymes are present in every sample, but their abundance is differing. That's why they still can separate from each other. So this is just a list of those genes where we found out body site specific pathways. So interestingly, this oral cavity had a high number of signaling molecules and interaction pathways. And there are many other observations, but yeah, so due to lack of time, we may not be discussing that. So I just want to know whether it is too much for like a pharma students, or should I like wind up with some conclusive.
No, sir, it's okay, sir.
So next, what we did, we tried to find out the functional cooperation. That means whether these bacteria listed here, these are top 24 species, which are on average at present at like 10% of the total bacteria inside that metagenome or microbiome. And like sorted this in the descending order until there is like fair number of percentage of bacteria present. So this only single species dominate all the metagenomes or microbiomes. So what we tried to do was map their functions to the metabolic pathways. For example, here the thymine biosynthesis pathway, uh, the red star is showing the absence of this gene, and these K numbers are the K IDs, which is assigned to a particular enzyme. So blank and red star means that the gene is absent in this particular species, Allistotypes or Bacteroides vulgatus. But if and it is also incomplete, this pathway is also incomplete in some other species, Eubacterium rectale, indicated by this red dot. This enzyme is essential for this function. So when we combine both of these pathways, the total complete pathway becomes complete. It is all green. So that means thymine biosynthesis can be a multi-step process where like different bacteria contribute different enzymes to carry out this for us. So then the outcome is the vitamin that we can use for ourselves. So what we did was we tried all these species and all these interactions in a network kind of thing, where this source bacteria contribute toward the direction of this arrow, where the target bacteria has this pathway incomplete and the source bacteria tries to complete that pathway. So these are all these such cases where we try to map everything to everything available, so that we know that which species contribute to the metabolic activity of another species or how they cooperate. So this is what I told in conclusion. So I would like to thank for my PhD supervisors about like providing their mentorship and resources by CSIR, also Calcutta University. Next, if there is still time, I can just go through quickly what I did in my postdoc at Germany.
Yes, sir. Please,
Okay.
sir.
So what we did was, so in 2015 or 16, there was a major uh, the report that indicated that the bacterial tree of life, or the tree of life, has one particular clade which is very abundant, and like there are too many number of species present, which is called as candidate uh, phyla radiation bacteria. So this tree branch is called candidate phyla radiation. So all these red dots means they are present in those environments, but the red dot indicates that they have never been cultured in the laboratory or petri dish. So what we are telling is only on the DNA basis here. So it is known that the CPR bacteria, candidate phyla bacteria, also renamed as Patexi uh, in today's literature. So they have very small genome size, like even below zero point 0.2 micron, and all other bacteria have higher genome sizes. So that's what that's what intrigued me to look into these bacteria. So it was known for this bacteria that they are either endosymbionts or fermenters or episymbionts, which is connected to some other body host cells or bacteria or parasites also. So our question was to investigate whether this genome size is somehow linked to cell sizes or not, and whether these bacteria form co-occurrence with other bacteria, so that we can say that these are probable host of these parasites. So what we did was, in the German forest land, we chose one plot and a forest land to sequence groundwater from the wells. So there were groundwater wells available on this hill slope. So it is going down, and in the agriculture, so it reaches in the agriculture area from forest land. So all these groundwaters were sampled, and metagenomic sequencing was done for microbiome analysis. So we did this complicated bioinformatics pathway to uh, come to the max, which is metagenome assembled genomes. So genomes of bacteria from this whole environment. So we come up with like hundreds of these bacteria and their subgroups of candidate phyla radiation bacteria. So it was all computationally done. So the major observation was, like, whenever the groundwater was filtered with zero point 0.1 micrometer filter, so the smaller cells were retained there in the supernatant. So these cells were sequenced, and we found the genomes from those metagenomes to know conclude that indeed the cell size, where the cell size is higher, the genome size of this bacteria also correspondingly increase. And then we did a co-occurrence analysis, where we show that these Patexi bacteria, which is red dots, can form a cluster with different number of nitrospira or other bacteria, so that they can hop on their resources and like use their resources for their own survival. So the exact names and other things were also published in this paper, but I think it is too much to discuss here. Then yeah, so the one more observation was, these bacteria had type 4 pilus protein, which is essential for attachment to different hosts. So, and similar observations were found that I discussed as a conclusion. So in the next step of these bacteria, we try to find out where these bacteria are coming from, either from soil, and they slowly seep in or drain into the groundwater. So we try to investigate whether this change in the habitat has some role or effect in their on their genome sizes. So as you can see, the genome size of CPR bacteria is falls within the range of all other obligatory symbionts from this, uh, even from groundwater, the red dots, and the range is also true for the seepage or drainage, which is water coming from soils. So they fall in the same range. Suggests that this might be also they might be also a parasite or endosymbiont of some bacteria. So we did the same in the same plot we did, but the sampling was done superficially below ground, like some centimeters below ground, to get this water. And this is the major source of this groundwater, where rain or snow melt events can contribute to the groundwater. So we use a lysimeter kind of apparatus to probe this water from soils, the moist soils. and we did the same kind of metagenomic bioinformatic analysis to get the genomes of bacteria. So we found out that so there is indeed effect of transition from soil to groundwater. So from brown data set to blue data set, the genome size is significantly changing for these parcobacteria, which and saccharibacteria, which is a class of candidate phyla radiation bacteria. So there is an effect of transition, and as they move from soil to groundwater, the number of pseudogenes, that is incomplete genes or false reading open reading frames, were higher, indicating that there is an evolutionary effect which like forces them to lose genes, because pseudogenes means it will be losing genes in future. So that will shrink their genome further. So these were very interesting observations, and we compared the like presence of functions or molecular features in two genomes which were very close from groundwater and seepage, which is soil-like water. So all the red things are specific to the soil bacteria, including some antibiotic-resistant genes, and the blue ones are unique to groundwater bacteria, where there is like a function which is used for degradation of nitrate pollution in the groundwater. So from the agriculture, some nitrate is contaminating the groundwater, and these bacteria try to utilize that nitrite, and reduce that to a safer byproduct. So these are the same conclusions that I discussed. So I would like to thank my postdoc supervisors, collaborators, and all the lab resources that were provided to me. So currently I am working in the Geo Institute at New Bombay. So we are focused on microbiome-based therapeutics. So as you might be knowing that there is one US FDA approval for a drug which is not actually a drug, which is a fecal transplant, which is a fecal material from a healthy donor, which is condensed into some kind of formulation and then transplanted into the disease one. So mostly in inflammatory bowel disease patients or Clostridium difficile patient where the drugs do not work. So there are therapies which are now microbiome based therapies. It is like a probiotic, but is a bunch of probiotic coming from somebody else's gut and then like transplanted to your body. So there are very like good opportunities for connectivity and industrial exposure in Geo Institute. So if you are interested, I can share also the details with you, sir. And the salaries are like for the PhD student and research fellows are more than the government salaries. So thanks all for your attention. And if there are any direct questions or simple like clarifications from my presentation, I am happy to answer.
Thank you very much, sir, for wonderful talk. I hope everyone present here has some valuable information to take away from the talk. I wish you very, very best, sir, and a wonderful day ahead. If anyone have any inquiry, I think there is no inquiry, sir. Thank you very much for your valuable time, sir.
Yeah, thank you.
My job भी निकली हुई है.
Rajbi sir, lecture is completed or not?
Sir, it's completed, sir.
Okay, so you can stop the recording and conclude the session. After conclude the session, you can stop the recording.
Sir, I already concluded, sir.
Okay, so you can stop the recording, sir.